Good morning, everybody. Welcome to Science Saturday. I am so excited to be here in Frederiksted, St. Croix. Woo! And this month we are talking about the stony coral tissue loss disease, which is following up on what we did last month, but we are today looking at it from the standpoint of the strike teams. Ooh, accidentally covered the camera there. So we are going to flip around the screen to our favorite scientist, Joe.
Hey, look everyone, I'm out of the studio. I'm here in St. Croix as well, which is really exciting. So last month we were talking about stony coral tissue loss disease. This is a disease affecting all three of the U.S. Virgin Islands, St. Thomas, St. John, and St. Croix. And so we wanted to go to St. Croix to talk with their strike team specifically about what we're doing to combat this disease. So just as a little reminder, we talked with Sonora last time about what that disease is and what's going on with it. Now we're going to be talking with folks who are actually getting in the water and working to prevent that disease, which is something really exciting. Again, this is something that I've been personally working on for a long time, Kitty as well, and so now we're going to get to see the people who are in the water with us. So why don't we go around and see who we have with us today. So first, I wanted to introduce Mike Funk.
Hey.
Mike has been living in St. Croix for a while now, Mike, and you've been involved with the disease response for quite a few months now, right?
Yes.
And you've been with and you've gotten involved through the CORE Foundation, which we mentioned.
Let's show that logo.
Yeah. We mentioned it before, but CORE has kind of been the group that's largely responsible for implementing these actual disease treatments. We also have with us Matt Davies.
Everyone.
Matt's actually from the East End Marine Park, but also involved through CORE as well, working on the strike teams, right, Matt?
That is right, yes. I do the coordination of the strike team here in St. Croix.
Right. And since we can't have Mason with us today, who do we have with us today, Kitty?
We have this month's Science Saturday intern, Kenji. Hello.
I'm just gonna be someone here. Probably gonna ask questions about the body of coral and what the disease actually does to the tissue.
Ooh.
Very good, yeah.
You are ready. That was not rehearsed at all.
And it was just his birthday. So.
Yep. So we are all
Sorry, I
about
had to say
birthdays
that.
on Science Saturday. My birthday was the day after Science Saturday in January.
That's true.
We should just do a birthday party every month. That's it. No more
Agreed.
science, only birthdays.
And we'll talk a little bit about birthdays later on. But for now, why don't we, Mike and Matt, can we just review a little bit about what the disease is? We have a little bit, something we didn't necessarily have last time, these are actual coral skeletons, right?
That's right, yeah. So these are some skeletons that were unfortunately affected by the disease. And in the early stages of the disease outbreak on St. Croix, and it was limited to quite a small area, we were hoping that perhaps if we remove some of those sick corals from the water, we could stop the spread of the disease so much. So you can see here on the skeletons of these corals, you can actually see this very distinct line. So this was all of the dead area, and the disease was slowly progressing across the coral, and this was all of the living tissue on this side.
Oh.
So Matt, Kenji had a question about that actually.
Yes.
Does the disease take away the color and life effect of the inside and around the body?
Yes, so actually what kind of happens is the living tissue on the coral kind of like melts away.
Ew.
So what you're left behind with is this bright white skeleton, and then this would have had the living tissue still on it, and it slowly moves across, sort of melting the tissue away. So that's why it's called stony coral tissue loss disease.
So,
Kind of floats away.
based on other Science Saturdays we've done about coral, we've learned a little bit about the biology of corals, and that color comes from the living organisms in there. So what we're seeing is the loss of color is coming from the death of that tissue, right? Am I saying this right?
That is correct, yep.
Nice. Yes.
Nailed it.
Nailed it. I pay attention during my Science Saturdays.
Well, coral is a symbiotic relationship. There's the coral animal, and then there's an algae that lives with the coral, and that's really what gives the coral its color.
Right. Awesome. That's a really good point.
Supplies a lot of the nutrition for the coral.
Okay, so this isn't quite on topic for today, but while you're talking about the relationship and that algae inside giving the color, I think that we can talk for 30 seconds about coral bleaching and how that's different from the tissue loss disease. Who wants it?
Sure, yeah. So this, I'm happy to take this question. So when we talk about coral bleaching, what's interesting is kind of what Mike was saying. When we see coral bleaching happen and not coral disease, the tissue is actually still on the coral. So if you actually go onto a reef and you see a coral like this and it looks all white, sometimes if you look a little bit closer, you'll actually see there's still a little bit of soft tissue. And that's the part of the coral that's actually alive. The rest of the coral, this rock area, that's the coral skeleton, you know, like the bones that we have, but the actual small bit of tissue is where the coral's alive. And what's interesting about coral bleaching versus this disease is that when we have coral bleaching, that tissue will still be there. It's just very, very clear and can be very hard to see. Sometimes it's actually inside these little ridges and valleys of these brain corals. So you see how you have all these teeth in here? Can you see these like teeth in here? So the corals will actually just retract way into these valleys in here, and so it can look like it's all gone, but it'll actually come back if the water temperatures go down or whatever's stressing the coral goes away. With this disease, unfortunately, that tissue is gone. It's completely gone. That part of the coral has died. So you can imagine, as Matt was pointing out with this line, one side of that line is dead coral skeleton, the other half is alive still. And so that's kind of what we're talking about with this disease, right? Is we have the corals that are we have this disease that's slowly moving this line across the corals, and once that line reaches all the way around the coral, that coral is going to be completely dead. But if we can stop that line from progressing, then that coral we can effectively keep that coral alive. That coral can keep growing, even if it's not the whole coral, part of it can keep growing. It can regrow that tissue, and we can actually save that coral from being lost to this disease. And that means more reef structure, that means more fish, that means more habitat, more beautiful things for us to see when we go into the water. All of those things are why we want to keep these corals alive, or at least keep as much of them alive as we can.
Right, coral is super important, and I know that we've talked about this in a few of our Science Saturdays, but just for those who are not obsessively watching us every month, which, you know, I'm sure there are a few of you, you know, we've talked about a lot of reef benefits, such as I can't remember what month, maybe it was December, we were talking about the shape of coral and how they grow together to protect the shoreline. We have fish who live within the coral reefs, really important reef species that are important to us economically, for food sustainability. So really important stuff and very good reasons for us to protect the coral.
So, on that note then, how do we treat this disease? And I think what's interesting about this is the way we treat this disease is actually kind of similar to how we treat diseases in humans, right? So what we found, or what scientists across the Caribbean have found, is that using antibiotics, so special compounds that are killing off bacteria, are really effective at stopping this disease from progressing, right? So, Mike, do you want to talk a little bit about like what antibiotics are and what we're seeing with the corals and the treatments there?
Sure. So my background is in medicine. I'm a physician assistant.
In human medicine?
Beg pardon?
In human medicine.
Human medicine.
Right.
Ooh.
Now he's treating corals.
Well, they're animals too, but in any case, antibiotics are very interesting. The first antibiotic that came out was penicillin, and that was in the 40s. It was found by accident. They were growing some bacteria, and some mold flew in the window and landed on it and killed the bacteria. Wow, that's really interesting. And they were able to identify the chemical penicillin and use that as an antibiotic. And they found it cured most of the diseases that were killing people in that time. Mostly upper respiratory tract infections, intestinal infections, and things like that. And then over time, they've developed other antibiotics. They found other types of molds and fungus and things like that, and been able to extract the antibiotics. So a lot of times you'll hear people talk about a strong antibiotic. That's really not a description. It's better to talk about a broad spectrum antibiotic. So broad spectrum means that it covers a lot of different types of antibiotics. And what we found is amoxicillin seems to work the best. It's what we
For
found so far.
for this disease.
For this disease.
On the corals, right?
On the corals,
On the corals.
yeah. I'm still thinking about mold flying in the window and that whole story, but we're gonna stay on topic.
Okay.
That's why people are here with me, because I'm just off,
Yeah.
in some cartoon in my
And I
head.
could go off on antibiotics. I mean, we have cephalosporins that were found in a fungus that's in sewage. So anyway, amoxicillin works when bacteria multiply. They split in half and then rebuild to make a full bacteria. In that process, they're building what's called the cell wall.
Oh.
Amoxicillin interferes with the rebuilding of the cell wall. So all of a sudden they don't have like an outer skin like we have,
Oh.
and everything just dissolves away.
This is a level of knowledge on antibiotics I never thought we would achieve. So this is awesome. So how does that translate then to the treating of the corals? I know you brought some stuff with you today. What do we have going on here? We've got a big tub.
So your question is a very, very good question. How does it work? And with a lot of the answers that we have in medicine, we don't know. We don't know exactly what the pathogen is yet.
Right.
All we know is as we tried it, seems to work.
Okay.
Right. And that so this presents kind of a unique challenge. We have this human medicine piece, amoxicillin, that we want to put on corals, which grow in the water. We can't we can't prescribe them a pill of amoxicillin.
Right, they would not follow the instructions.
Unfortunately, no, because as we've maybe talked before, corals are actually a bunch of individual little upside down jellyfish growing all over this skeleton with tiny little mouths. So we got to figure out a way to get this amoxicillin to the corals and get it so that they're getting the right amount of amoxicillin, right?
And over time as well.
Right.
It's not just one shot.
Right, exactly. And
So dosing.
Right. So we have to. So as Mike's saying, with antibiotics, because we have to make sure that we're, getting all of the bacteria that we're trying to get off, and saving some, of course, but we need to make sure that it's done multiple times. So this is really common in human medicine. If you take an antibiotic, you have to take it, once a day for several weeks. So how do we do this in corals rather than telling them to take a pill once a day for several weeks? This is actually something that some folks up in Florida started working on. This is a group called Ocean Alchemists. I think you have them right here, right, Meg?
Ooh.
So Ocean Alchemists has developed this specialized paste that actually goes onto the coral, and over time, using the water movement all around the coral, washes that amoxicillin right onto the coral skeleton and provides that antibiotic dosing. So there's a little bit of it in here, Mike. You want to get some out of it there?
Oh, I smell it. Woo!
Yeah, it's good. And you see it's real.
Yeah,
that's
here's
why I'm wearing-
Kenji.
that's why I'm wearing-
Here, we can take a little. I'll take a little bit of it here. Just.
A little treatment.
So this is a little treatment. You want to feel some of it? You want to get some there?
You want to touch it?
Once you
No,
get it-
looking like it doesn't want to touch it.
It
This stuff
washes off with
this
soap
stuff is
and water
totally
a little
this stuff is totally safe for humans to touch. This does not contain any of that antibiotic. This is just a paste, and it's designed really just to stick really well to corals in the water. It's not attracted to the water. It's hydrophobic, so it's not necessarily going to stick to the water. But as you can see, it's pretty good sticking to a lot of other things. And so that's what's really cool is we can actually put this paste on here and then leave it. And then by leaving it over time, these corals get this treatment. So this is really useful when we're going out and we're treating corals and we have this line, right? The coral's not totally dead yet, and we want to stop it from advancing. Well, we can put that antibiotic, I'll get a little bit here, a little antibiotic, and we put it along this line, and we can stop that from progressing, and we can keep part of that coral alive using antibiotics, using human medicine in the water, which is really cool.
And we can demonstrate that.
Right,
Yeah,
make
so
some stuff
we've
up.
got the
we've got
the base, we've got the medicine, and you guys mix it together and then take it out on the reef with you?
Yeah.
All right, is this a demo time?
So I think so. So
All right.
We don't have any living corals with us today. Unfortunately, we're still working on getting underwater for Science Saturday. It'll happen, I promise. But, in the meantime, just because because we get this stuff for coral treatments, and we're not going to actually treat these corals, we're going to use something that's very similar. Right here.
Oh
this
man,
is something
the
that
power
we've
tools
used
are
in the
coming out.
past. Shea butter, actually. And this is actually something that we can mix in with powdered white amoxicillin. So, and we're not going to use amoxicillin today. We actually just, as a very similar in how it looks, this is a red flower. So here, you guys want to feel some of the red flower here? So this is very similar to how the amoxicillin feels when we receive it. It's just powdered white. Red flower. You can see that's still getting everywhere, but if we put that on without the paste, it obviously wouldn't stay on. So we can use a paste like base to be, or in this case, we're going to use shea butter. And Mike, Mike or Matt, if you guys want to show how you would mix that up.
All right, Kenji, you're going to do it next, so get in there and...
Okay, so I'm going to show you. So we actually measure it out. I have a scale, so I usually, if we're going to go out on a dive, I'll take 400 grams of the base 2B and 50 grams of amoxicillin, so it's got to be fairly accurate. And you scoop it out, and I put it in here and measure it up. Go ahead, scoop a bunch in there. Yeah, good.
Oh yeah, get that shea butter. It looks a little like ice cream, yeah. After Science Saturday today, we're all gonna do some self-care moisturizing. Yeah, maybe not with the flour. I don't know what benefit that would have. Okay. Nice. Looking good.
Okay. Now why don't you put a couple of scoops of flour in there?
Yeah. You ever help your mom baking, Kenji?
That's good.
This is good practice for baking.
Oh, what you got there, Mike?
Whoa.
So this is what we found is the most effective way to mix the stuff up. So I sacrificed my egg beater, and I'm using a portable drill, and that works real good as like an egg beater. And I just put that in there and mix it up.
So just to be clear, we have combined human medicine, coral biology, and baking and power tools. Just in case anybody was keeping track of our cross-topic work. Man.
Now, as you can see, it goops up my my egg beater.
Right.
So I got to scrape it off of here with this.
I just want to point out that I was helping getting ready for a treatment dive a few days ago, and I was not given an egg beater with a power tool. I used a knife and did it by hand. Not even a spoon. I did use a spoon to put it in the syringe, but...
Okay, so the first time I ever did it, I was given a teaspoon with a handle about that long. It fit in my hand like this.
Oh no!
And it was torture.
The sacrifices that the volunteers of the Core Foundation go through for the treatment of our corals is astounding.
Well, we try and figure out ways to make it easier.
Right, yes. Try to make it
Okay.
easier.
So this is actually a garden 100 ml syringe, and I cut the tip of it off. So I have about a 1/4 inch diameter opening in there. And then what we do is,
I would like to see Kenji trying that.
yeah, I'm going to show you how. You
fun
grab
part,
a
it's
little
the
bit
messy
of the
part.
stuff in here, and you push it in with the spoon like that.
All right, you ready, Kenji? I see you studying.
The challenge is to get it all in there without getting any on outside of the zoo.
Right, we want to try and keep it outside of it.
So while you guys are working on this, I think this is a great time to say that if you would like to support the efforts of these amazing volunteers who are sacrificing egg beaters and spoons and putting all of this work, you can make a donation directly to the CORE Foundation. And their website is corevi.org. You can also get there from the VI Coral Disease website.
Push it like this.
Man, this is...
Mike and Matt, how often are you guys doing this, and how much are you guys generally using per week? Or
That's a good question. We try and get out to each of our treatment sites every 2 weeks, and we currently have four treatment sites, but we're probably going out, probably doing at least three, 4 dives a week. We use about 400 grams of the base CB and amoxicillin mixture on each dive. Although it depends how many divers, because sometimes if you have 4 or 5 divers, we'll use almost double that.
It's a lot of work.
Yeah, it
A lot
is.
going into this.
Yeah, Mike's a real star out on his boat. Probably every other day, huh?
Gotta be.
and all the
Okay, so we
volunteers.
got that about, we don't fill it all the way up because we have to be able to put the plunger back in. And then, when you squeeze it, a little it comes out like that. That's not going to hurt you. It's biodegradable.
Again, this is all very safe, environmentally friendly, and this is our shea butter flower version. So
Right.
no animals or plants were harmed.
Can I demonstrate how we put it on?
Yeah.
Okay, can you watch? Because you're going to be doing this, okay? So what we do is we squirt out a little bit like this in the water, and then you want to try and anchor it right where that disease lesion is, and you got to mush it down into the coral.
And why do you have to mush it down, Mike?
You got to get all the way inside of there, so you're going to build basically a wall where the disease can't get through. If it's not all the way down inside of there, the bacteria or whatever it is can get underneath. So you got to get right down inside of there. And you want this to be about 1 centimeter wide.
Nice little treatment line.
Nice little treatment line. And it'll get away from you, and damselfish will come up and steal it from you and all that.
Damselfish, the true dangers of the ocean.
They're so cute, but they're pests.
So also, it's important to remember these guys are doing this underwater in dive gear.
Right.
So I think that we brought some stuff to kind of demonstrate that, right?
So
Let's get Kenji geared up.
All right, so
Okay, all right, you get geared up. I'll
Kenji, do you want to
continue.
get your dive gear on?
all right, so today Kenji is going to do our coral survey, looking for corals to treat. Got this awesome setup. Mask up. And for breathing purposes, we're gonna let Kenji take his face mask off and just stay 6 feet away, because we're safe. All right. Are you ready? Yeah? Okay.
You gotta go search around for a coral that looks diseased.
All right, let's go find a coral. I hope it's not Sergi. Whoa. Whoa. Where are you going? You passed the coral.
The reef's over here.
Oh, there's one.
So it's really common to treat brain corals on the reef. So we're on a place that looks like there's a lot of brain coral.
Uh-huh.
Do you see the disease line there? So they look right along here. Yeah, it's kind of hard to push, isn't it? Here, I can help too. Your dive buddy will come in. Push that.
It's really hard to push.
Yeah. Here, I can push that, and then you want to push it in? Okay, ready? Here we go. There you go. Make sure we're getting right into those walls.
So when the strike team volunteers are out in the water, you do have teammates and fellow volunteers to help out. So you're not on your own, and you can make sure that the treatment gets in there nice and secure.
There you go, pressed in there.
All right, looking good.
We got to make sure, and we want to make sure we treat that whole lesion all the way to the end, right?
And if I could say a word about that core, we require all of the proper diving safety measures. We do buddy system where we check everybody's air and do all those things as we're going along.
Yep, because we are safe and we are protecting the reef.
So, looks like we got this side.
You see a little bit right there.
Come over here. All right.
If you don't get the whole thing, then that disease can just sneak through the gap.
There we go. And nice and mushed in there. How's that? You think it's pretty good?
Pretty good.
Looks like there's a little spot right there. You ready? Let's get that a little bit.
All right, and so if you guys were out on the reef, you would also be taking a picture and documenting this whole thing, right?
Right. Yeah, so we don't have a camera, so all right, next coral. Come on. We gotta treat all the corals. Here we go.
Let's do that one in the shade.
Okay.
There's one.
This coral is a little bit different, right? This is a starlet coral, Siderastria cinerea. You want to try pushing and I'll press it this time? Oh man, this is a hard one because it looks like there's a lot of different features, right? I already want to switch. All right, that's what buddies are for. Here we go. I'll get neutrally buoyant here.
Make sure your fins stay off the reef, Joe. This is quality content, guys. You won't get this anywhere else.
You're doing great. We gotta get you trained up to start diving with the strike teams for real. Okay, wait, where am I going next? Over here? Down here?
Strike team.
We have a Facebook page, the STX Strike Team. So you can send us a private message on there. Make sure to like or follow the page. We post lots of updates about how many corals we've treated, information about the different species of coral and about the disease itself. And you can shoot us a message on there or leave a comment on one of our videos. And yeah, that's probably the best way to get in touch. We do have a very comprehensive training process. We have a 2-hour virtual training session we invite our new volunteers to go through before we first get them in the water and continue our training there before they move on to finally getting in there and treating the disease.
Right, for the good stuff. And as you can see by Joe's buoyancy techniques, you do want to make sure that you're, a qualified diver, comfortable in the water. You can start doing buddy dives and helping to find the coral and taking photographs. But to roll around upside down and treat, really expert level stuff here.
Kitty, do you want to talk about the reporting form that we have? Because that's something anyone can do.
Yeah, absolutely. So if you are a snorkeler or a diver or you're out swimming and you see coral, I'll switch around to my face for this, although this is really fun to watch. Hey, so if you are out snorkeling or diving and you see coral that just looks kind of weird, you can report it on the Coral Disease Advisory Committee's Coral Health Report. So to get there, you're going to go to the website, which is vicoraldisease.org. And there's a button on the top of every screen that says Submit Report. And from there, you can mark down where you are, make some notes about what it looks like, submit photos. There's a little pin dropper to show where you are. And the photos come to us at the VI Coral Disease, and we will figure out what's going on, look at the pictures. You can also report a totally healthy reef, which we love. So if you're out and you see a reef that is perfectly healthy, definitely no coral disease, everything looks good, you can submit it as an all clear. And that is great for a few reasons, because the data is really important, that we know where the disease is and isn't. And also for us, looking at pictures of dead coral all day can be really depressing. So we love to see the healthy coral. So we're going to check back in with our dive team. It looks like they've moved on to another coral. How are you guys doing?
How are we doing, Kenji?
All right.
He says we're doing pretty decent, actually.
Pretty decent. Awesome. I love it.
So you're noticing, Kenji, a difference between these brain corals and the star coral for pushing it in? How we have to really get into these ridges, right? Yeah. Coming along here, though. And so, Matt and Mike, we usually, when we treat these corals, we're usually treating them once, and sometimes that'll stop them, but a lot of times we have to go back and re-treat these corals, right?
Yeah, unfortunately, this doesn't stop the corals from getting reinfected. So sometimes what will happen is, even though our treatment works and stops that lesion from progressing across the corals, what can happen is a new lesion might start a month or two down the line. So we go back to our sites and we check on the corals that we've already treated and make sure that we're giving them little top-ups as and when they need them.
Cool. Very nice. So we're at 30 minutes now, and we've got a little walk down memory lane we're going to finish with. But I want to give you guys an opportunity to kind of finish off. What are your final thoughts on what you want the public to know, specifically about the coral disease and the work that you're doing?
Well, from the core perspective, we're looking for volunteers that want to help us, and we're also looking for the treatment process. The amoxicillin, the base 2B, is relatively expensive, so we need money to buy the product. So we need help. We need divers in the water, reporting, and we need
Like this
funds.
guy. All right, awesome. Anything you want to add, Matt?
I think we're going
Yeah, I just sort of echo what Mike said, particularly about the coral health report form that Kitty so eloquently described earlier. That's a real help for us. You know, we are somewhat limited in the amount of time and people that we have that we can get on the water. So the more eyes we have on the water, the better. So if you see anything, we really appreciate those coral reef health reports.
Awesome.
They're a real big help to us.
Yeah, I will second that as somebody who just kind of does the outreach and communication for a lot of this. Being able to get reports from anybody in the territory, I think, is really helpful. Absolutely. Kenji, what is your final thought on what you learned today? What do you think about this stuff?
I think it's
Pretty decent, but it can be a pain to try and actually spring around if you're the large kind.
Right.
Currently going around the area.
Right, yeah, that would be much harder if it was like current pushing you back and forth or surge or something.
Yeah, I think I can follow that. It just, it means that we can't necessarily treat every single coral this way, but we're always looking for new ways that we can treat corals. We can help restore corals after they've been diseased. So we're always looking for new ways that we can help, work against this coral disease. And there's a lot of things that, we can ask, for everyone to do to make sure that we're taking care of our reefs. Of course, there's things like what we were just talking about with reporting anything that you're seeing that's affecting the reef. That's super helpful. And in fact, one thing we were just talking about is, following things like the Strike Team page so you can learn what's happening, where the disease is. So those are all really great ways to help the reef. Also, just keeping in mind, there's lots of other things we can do to help the reef so that they're not suffering from other things while it's being affected by disease.
Right. I think that's a really good point. A lot of people ask, what can we do to stop the coral disease or, help the corals? And giving them the best chance at health, I think, is really important. Sorry, blocking the screen there. I'm going to move into the shade so my phone doesn't shut down.
One last shot of all the corals that's treated here.
Ooh, yeah. So,
Reef looking.
Reef is looking good. Good job, guys.
Great job, Candy.
All right, so I want to finish today with a little trip down memory lane. Today, or this month, is the 4-year anniversary of Science Saturday. And in those 4 years, we have only missed 1 month, and that was September 2017 for very good reason. It was a very windy week, and we just won't go more into that. But I wanted to take a minute and give an opportunity for us to talk about just appreciating and celebrating that Science Saturday is still going strong. And I thought that we could share some favorite memories, maybe throw a few pictures up in the comments. So I'll start by saying, Joe's taking it.
I'm gonna take over the camera here since Kitty, we're gonna be pointing it at you.
All right.
Yeah, so Kitty, you've been doing this for 4 years now.
Yeah, this is our 4-year anniversary this month, and we started in person, obviously, at American Yacht Harbor on St. Thomas with the help of Lattes in Paradise. And I really miss having my coffee and Science Saturday every month, but I was able to swing by Polly's and pick up a coffee this morning. But I just want to thank everybody who has helped me keep it going. I've tried really hard to make it consistent. We never cancel, we never move it, except for that one September. And it's been really amazing to see the families who have stayed with me this whole time, you know, who started coming in person and are still watching us on Facebook Live and have helped me and provided supplies. And I just wanted to point out a few of my favorite memories. One I really want to point out was actually October 2017. That was our first Science Saturday after Hurricanes Irma and Maria. I didn't know if we could do it, but I didn't want to cancel. And so we got seeds donated from Jenny from Lattes in Paradise. We got soil, and we just did planting, and it was really, really cool. And we kind of celebrated rebirth, and I think it was really fun for everybody to get back out. Oh goodness, we've done so many really cool ones, and have had so many amazing guests who have come to help out. We have had other DPNR divisions. The university has been a huge help, and I think that one of the real great things that we can celebrate is when Joe started helping. So,
Hello.
we're going to turn it back around.
I feel like we're going back and forth, but yeah,
We
Kitty,
can go this way a little bit.
I've really only been helping for about a year and a half, but this program, we're always looking for new content and new scientifically interesting content. What we're always talking about is, how are we bringing science and how are we using science in the real world? How are we using science every day? I mean, today we learned about human health and coral health all in the same program. So some of my favorites have been things that, although I've been working as a scientist for a number of years now, things that I've never studied in my scientific study. Like for a time we did have had Jason also from the Core Foundation, and we dissected lionfish.
And we ate it.
Right.
I made ceviche right there in our yard.
Right. So one thing I think would be great if folks who are watching, if you guys have any photos or anything from old Science Saturdays you guys want to share, feel free to share in the comments here. Talk a little bit about some of your favorite Science Saturdays. I know we've got some regulars watching. I know personally, I'll share one of my favorites was actually one of my very first Science Saturdays. It was before we were recorded, unfortunately. But I'm happy to share the activity with anyone who's interested. It's teaching all about ocean acidification, and we actually used cabbage juice, right, Kitty?
Yes.
Right.
It was delicious.
So using the cabbage juice, we can actually bubble carbon dioxide from our breath into the cabbage juice and use it as a pH indicator. And so I'm happy to share that activity with anyone who's interested who wasn't able to be at that Science Saturday. But we have all sorts of activities like that that are talking about, real science that scientists are using and how we can see that happen in our everyday world. So that's one of my personal favorites. It also got very inquisitive when we were looking at what other things might affect the pH of that cabbage juice.
Yes, you guys went into the sun, you sunbathed with it. We had some participants add soil to it. Really extreme bubble making. That was a really fun one.
Kitty, do you have a favorite virtual Science Saturday?
Oh man, the virtual one. So
we're marking about a year. Next month, I think we'll mark a year of our first virtual Science Saturday.
Yeah, so we've been doing virtual for a year now. I thought that back to me. Whew, my hair is okay. So I've been really impressed with the people who have been joining us and helping us. And you know, you mentioned the lionfish one with Jason from Core, and we got to eat it, and it was delicious, and we had some really fun comments on that one. We had a PETA representative, which was super fun. This one today, to be able to host on St. Croix, I think is so exciting. I'm really happy to be here. I want to go back to before we were virtual, and I have a photo of this one that I can post in the comments, but Joe's first Science Saturday, we were talking about bats, and we have a picture of him sitting in one of the tiny chairs in the rain cutting out a bat mask, and I thought that was pretty fantastic.
Well, Kitty, we do have a comment. Marilyn did want to mention, congrats, Kitty. They have all been amazing. She has too many to choose a favorite.
Well, thank you. Marilyn, I will say that the amazing questions from the stay-at-home scientists is always appreciated. Liam and Hazel are some of our favorite fans. We love you all. And I don't want to point anybody out, but I will